Okay, am I audible?
Yes, sir, audible.
Okay. So today we are going to see glycolysis, right? Okay, glycolysis. हे दिसतंय तुम्हाला पॉवर पॉइंट दिसतंय? Is PowerPoint visible? संकेत पॉवर पॉइंट दिसतंय का? ओके, पॉवर पॉइंट दिसतंय का तुम्हाला?
Yes, sir.
Yes, sir.
ओके. See, people are hearing. Okay, so today's topic is glycolysis and gluconeogenesis. So when we say glycolysis, glycolysis means breakdown of glucose. Okay, under carbohydrates, there are different carbohydrates like glucose, fructose, lactose, galactose. Likewise, so we are going to see glycolysis. So first we are going to see what is glycolysis, and in the next step we will see what is different steps of the gluconeogenesis. So glycolysis, what is glycolysis? So basically glycolysis is a sequence of reaction that converts one molecule of glucose to two molecules of pyruvate with formation of the 2 ATP molecule. So basically we have glucose molecule with us. Still someone is running. Okay. Okay, so the purpose of the glycolysis is that we have one glucose molecule. Okay, glycolysis. Here we have one glucose molecule. Now this glucose molecule we have. And what we want to do with this glucose molecule, we want to break this glucose molecule into two molecules of the pyruvate. So ultimately we will be getting two pyruvate molecules, and we will be getting some ATPs here, two ATP molecule, adenosine triphosphate. So this ATP is the source of energy. So this ATP we will be getting, and this glycolysis can be anaerobic. Okay, so this is one can be anaerobic, means it can be remain activated under the anaerobic condition, means without oxygen also this reaction can occur. Okay, why glucose is such commonly fluid? So now whenever body uses most of times we see glucose is a source of energy. So why glucose is a source of energy? Because this glucose tend to form the ring. So if you see the structure of the glucose, it is in the ring form. It is very stable and it doesn't glycosylate the proteins. So this glucose molecule does not easily react with other protein molecule in terms glycosylating the protein. If this glucose as a predominant source of energy, if it is combining with the protein and it is glycosylating, it means that the protein function may get hampered. So that is why we have glucose molecule, which is a ring form and very stable form of the stable form we are using at a energy. So formed from pyruvate formaldehyde under the prebiotic condition. So this one is formed from the formaldehyde under the prebiotic condition, glucose. So what are the possible fates of the glucose? So we under unit 1 we have studied the what is carbohydrate. So this glucose. So C6H12. So this molecule C6H12O6. This is a glucose molecule. It has 6 carbon atom in its structure. And now when this molecule undergoes the glycolysis, what can happen to this molecule? Already we are discussing that this glucose molecule, it can give rise to the pyruvate molecule. So this pyruvate molecule, two pyruvate molecules can form from the single glucose molecule. Okay, so one fate of the direct fate of the glucose is that it get converted into the pyruvate molecule. Okay, so this glucose is getting converted into this pyruvate molecule. What happened next? This pyruvate molecule can have the three fates. We call it as a fermentation phase. Under the fermentation phase, this pyruvate molecule can give the ethanol, means it can give the alcohol molecule, or it can undergo conversion to the lactate molecule. These are the two fates. And third fate is that if it comes in contact with the oxygen, this pyruvate molecule can get converted into the carbon dioxide and the water. That is a complete oxidation. So basically we have glucose with us. Now the glucose molecule is there, this glucose molecule is there, and now we want some energy from this molecule. So what we can do, we can say this molecule undergoes the glycolysis and it forms the pyruvate. Okay, now pyruvate has formed, and this pyruvate can undergo in this reaction oxidative complete oxidation to carbon dioxide and the water. This is the fate under the aerobic respiration. And if there is anaerobic condition, under anaerobic condition, this pyruvate molecule can undergo conversion to the ethanol molecule, or it can undergo conversion to the lactate molecule. So these are the different fates of the glucose molecule. So what can happen to the glucose molecule inside the body? So what is difference between the these are the some questions like what is difference between facultative aerobes and anaerobes and the obligate anaerobes? So facultative anaerobes means they can function in presence of oxygen as well as in absence of oxygen. Obligate means compulsory non-oxidative conditions should be there. Means oxygen should not be present there. Okay, so what are the some examples of habitat dependent anaerobic or activity dependent anaerobic? So you can see under the rumen condition, some bacteria they are anaerobes. So they for their function they can derive the energy from the anaerobic glycolysis condition. Okay. So glycolysis. So all the intermediates in glycolysis have either the 3 or 6 carbon atom. Okay, so when we are talking about the glycolysis, we are saying that okay, glycolysis is there, and now in the process of the glycolysis, 6 carbon glucose is present. So number of carbon atom in the glucose is a 6. Okay. Okay, everybody is familiar with number of carbon atom in glucose, right? Ganesh, Borhade, Ganesh, Manoj Gauri. So basically, there are glycolysis can have either the 3 or 6 carbon atoms, right? So all the reaction fall into one of the 5 categories. So when we say glycolysis can have the 5 different categories. Okay. Phosphoryl transfer can be there. There can be a phosphoryl transfer. Second reaction can be like phosphoryl shift. So phosphoryl group is shifting within same structure. Phosphoryl transfer means A molecule is there and B molecule there, and A molecule is transferring phosphorus atom to this B molecule. That is a phosphoryl transfer. Next reaction can be isomerization. Isomerization means there can be some structural changes. Dehydration can be there, means some water molecule is coming out. Next can be aldol cleavage reaction. So these are the different reactions of the glycolysis. Entire reaction steps may be divided into the 3 steps. So we have this glucose molecule, for example, here, and we have this cell is present here. So what things are needed for glycolysis? So this glucose molecule, first it should get trapped inside. Okay, it should go inside this cell. So we call glucose is trapped and destabilized. Okay, so this is first step. Now glucose has come inside this cell. Now next reaction, this glucose, this is a glucose molecule, this is a 6-carbon glucose molecule. Now it will undergo this series of reaction, and finally these 2 molecules we will get. This is 1 pyruvate and this is 2nd pyruvate. So this glucose 6-carbon molecule gets split into the 3 carbon molecules is another fate. And what happens is that this is one reaction, this is second reaction, third reaction, likewise reactions is there. In these reactions, some ATP molecule we get here, some ATP molecule. Okay, so ATP molecule can also get produced. So ATP is basically, this ATP is a source of energy. So this ATP molecule can be we can get in the middle process. Okay. So now we say glucose molecule. So already previously we saw that glucose is a very stable structure. So you can see here this glucose is a ring shaped structure. Whenever there is no group shown, it means that hydrogen group is there. Okay, so this is the first carbon, second carbon, third carbon, fourth carbon, and the fifth carbon, and this one is a 6th carbon. If you see position of the glucose molecule, how it is, which groups are attached, here is a hydrogen atom, OH group, hydrogen atom, OH group, hydroxyl group, EH atom, hydrogen atom, hydroxyl group, and at the 6th position again there is a hydrogen atom here and OH here, CH2OH molecule. So basically this one is a glucose molecule. So when we are discussing glycolysis, the whole purpose of this lecture is that we have one glucose molecule. This glucose molecule is there, and now we want to convert this glucose molecule into the 2 pyruvate molecule. So how to convert the glucose into pyruvate molecule? So whenever any biochemical reactions is there, so we start with molecule. Okay, so this glucose molecule is there. Next, what things we need? So for any biochemical reactions, we will need some enzyme. So what is that enzyme name? So first enzyme name is a hexokinase. Does it make sense? Hexokinase enzyme. So let's see. we have this glucose molecule, we have enzyme hexokinase, and we have ATP molecule here, adenosine triphosphate molecule. Now, what is our first function? What we want in the first reaction? We want glucose to get converted into the glucose 6-phosphate. So, because this is in the hexokinase reaction, hexo means 6 carbon, and kinase means there is a involvement of the phosphoryl group transfer. Okay, phosphorus group is getting transferred from somewhere, one substrate to another substrate. So which is phosphorus carrying group? So this adenosine triphosphate is a phosphate carrying group, and from this group, this phosphate get transferred here. Okay, so we can see here is a formation of the glucose 6-phosphate. Now we will count number of carbon again. 1, 2, 3, 4, 5, 6. At the 6th number carbon position, phosphate group has attached. So that is why we call it as a glucose 6-phosphate. So now this compound we have kept, and this ATP has got converted into the ADP and plus hydrogen ion we are getting. So basically the purpose of glycolysis is we want some energy in form of ATP. But if you observe here, we are investing some energy. So glycolysis needs some investment of energy in form of ATP before it proceeds further to produce energy in the downstream process. So here what happened in the stage 1 of glycolysis is that glucose is getting converted into the glucose 6-phosphate. Here ATP is needed, and this reaction catalyzed by the hexokinase or the glucokinase. Okay, and induced speed is there. Induced speed means that this enzyme has a induced speed. So whenever substrate ATP and glucose is there, if it has to attach with the enzyme, there will be some structural changes in the enzyme. Then only the active site will be exposed. So that is why we call it as a induced feed. Okay. Next, free energy in the process is the minus 4 kilocalorie per mole. Now, in previous process, now we have got here glucose 6-phosphate. Now, what to do with this glucose 6-phosphate? Let's see what is happening with that glucose 6-phosphate. Here, this glucose 6-phosphate molecule is there. Okay, this is the glucose same molecule. 1, 2, 3, 4, 5, 6. So this is a glucose, and at the 6th position, phosphate group is attached. So that is why we call it as a glucose 6-phosphate. Now, this glucose 6-phosphate get converted into the open chain form. So this is a glucose 6-phosphate. So we can see this one was a circular closed ring structure. Now it has got converted into the open state. Let's count again whether it is same glucose or not. We will count number of carbon again. 1, 2, 3, 4, 5, 6. Okay, 6 carbon is there. It is a glucose molecule, and we can see here phosphorus group is attached. So that is this structure and this structure is the same, but it is just a closed circular structure, and this one is a open chain structure. So what happened is that during this reaction, the glucose 6-phosphate get converted into the fructose 6-phosphate, and the enzyme involved in this reaction is a phosphoglucoisomerase. So if you see, number of carbon atom are the same in this structure and this structure. So glucose 6-phosphate and the fructose 6-phosphate has a similar number of carbon, but just the arrangement is changing. Some arrangement is changing. So here we can see aldehyde group was present here, C11OH. So that is why we call this as a glucose. And here instead of aldehyde, this keto group is present. So CH2OH is there, and this keto group is there. That is why we call it as a fructose 6-phosphate. And for this reaction conversion of the glucose 6-phosphate to fructose 6-phosphate, there is involvement of the enzyme phosphoglucoisomerase. So this phosphoglucoisomerase enzyme is needed. Next, aldose is converted into the ketose. Okay, so this glucose molecule is an aldose molecule. We know that there are different type of carbohydrate, aldose and ketose. So this glucose is aldose, glucose 6-phosphate, and now it is getting converted into the ketose, that is fructose 6-phosphate. Okay, so the reaction energy involved is a plus 0.4 kilocalorie per mole. And now next step, what happen is that this open chain form get converted into this closed chain form. Okay, so basically this structure and this structure, both are the fructose 6 phosphate, but just the difference is that one is a open chain form, this one is a open chain form, and this one is a closed chain form. Okay, so this is a cyclic structure. Now we have got glucose got converted into glucose 6 phosphate with the help of hexokinase reaction. Next, in the next step of reaction, glucose 6 phosphate got converted into the fructose 6 phosphate. There was involvement of the phosphohexoisomerase enzyme. Now, what to do with this fructose 6 phosphate? So this fructose 6 phosphate is there. Now if we have this enzyme phosphofructokinase, in the presence of ATP, what happens to this fructose 6 phosphate? So this fructose 6 phosphate is there. Fructose 6 phosphate means this is a fructose molecule. Okay, this is, you count from here: 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, 6 carbon. You can see at the 6 carbon position there is a phosphate group attached, and now phosphofructokinase. So what is meaning of this phosphofructokinase enzyme? So we have fructose 6 phosphate, phosphofructokinase. So there is a fructose molecule and phosphate group is attached with this molecule. So you can see it is a fructose 6. That is why we call it as a phosphofructo. And why we call it as a kinase, this enzyme? Because in this reaction, at the third step of the reaction, this ATP, adenosine triphosphate, is adding the phosphate group to this fructose 6 phosphate. Now we have fructose 6 phosphate, and now we want to add phosphate group to this first carbon position. Okay, CH2OH group is there. Now we want to add our phosphate to this first position. So now this carbon is there. Now we are adding the phosphate here. So that is why we call it as a fructose 1,6-bis phosphate. So previously it was fructose 6 phosphate. Now we have added the phosphate which is present in the ATP. From this ATP we have added it here at the first carbon position. So that is why fructose 6 phosphate get converted into the fructose 1,6-bis phosphate. And now this ATP gets converted into the ADP, because one adenosine triphosphate has a three phosphate group. Once we remove one phosphate group, two phosphate group remains. That is why we call it as a adenosine diphosphate. This is adenosine triphosphate. After the reaction, adenosine diphosphate. So this after this reaction, this fructose 1,6-bis phosphate is formed, and this is a rate limit. This reaction, phosphofructokinase, is a rate limiting enzyme, and it can activate. It can get activated or inhibited allosterically. So allosteric enzymes are the enzymes which can get activated if some molecules attach with them, or it may get inhibited if some molecule attached. So how it is allosteric? So we call this phosphofructokinase as a allosteric, and this is rate limiting. Rate limiting means all the process of glycolysis will be limited by the what will be the rate of reaction. For example, there are the 10 step of the glycolysis, but this step is very slow. So what happens is that all the steps of glycolysis get inhibited, and the reaction rate will be similar to this rate limiting step. Okay, so now allosteric, coming to allosteric point. So we say that this enzyme can get inhibited. So who inhibits them? So this enzyme phosphofructokinase can get inhibited by the adenosine triphosphate. If adenosine triphosphate molecule is there, already ATP is there, then this enzyme may get inhibited, because already some ATP is there in the system. Now this additional ATP is not needed. It can be a cause of the inhibition. So next, citric acid. If more citric acid is there, this phosphofructokinase get inhibited. So we are showing as a negative sign, means inhibition. Long chain fatty acids are more, again negative sign. This phosphofructokinase can get inhibited. Now who stimulates it? Means we want fructose 6 phosphate to get converted into the fructose 1,6-bis phosphate. So in that case, it can get stimulated by ADP, adenosine diphosphate. It can get stimulated by AMP, adenosine monophosphate. So here we show as a positive sign. Okay, so this is a stimulation reaction, means positive feedback. So more ADP is there or more AMP is there, it means that this adenosine triphosphate, it will try to increase this rate of reaction. So this fructose 6-phosphate will get converted in the more quantity to the fructose 1,6-bisphosphate, and the energy required in this process is the minus 3.4 kilocalorie per mole. Okay. So till now we have seen this thing. We have glucose molecule here. Now with the help of hexokinase and ATP, this glucose molecule get converted into the glucose 6-phosphate. Here we have added the phosphate. So glucose 6-phosphate has formed. Now next enzyme in the process of glycolysis is the phosphoglucose isomerase. So with the help of the phosphoglucose isomerase, this glucose 6-phosphate get converted into the fructose 6-phosphate. So now this molecule has get converted into this molecule. So this was a aldose molecule, glucose 6-phosphate. It has get converted into the ketose molecule, fructose 6-phosphate. Now fructose 6-phosphate with the help of phosphofructokinase, it get converted into the fructose 1,6-bisphosphate. So this ATP molecule already fructose 6-phosphate was there. One more phosphate group got added here, and we got fructose 1,6-bisphosphate. So this is a when we say glycolysis cycle, there are glycolysis cycle is divided into two steps: preparatory phase and the post-preparatory phase. Okay, downstream phase. So preparatory phase means that the purpose of glycolysis is that we want ATP from the glucose molecule. But are we getting energy in this phase? Here, what is happening? ATP is getting utilized, ATP is not forming, ADP is forming. Here, ATP is getting utilized, ATP is not forming, ATP is utilizing. So though ATP is not forming in this first step, like hexokinase reaction or phosphofructokinase, but this is the preparatory phase. If you want more energy in the form of ATP from the glucose molecule, first you have to invest some energy. So that is why this ATP is used in the hexokinase reaction or the phosphofructokinase reaction. So these are the preparatory steps of the glycolysis. So now till now we have come to the point of the fructose 1,6-bisphosphate. Now what to do with this fructose 1,6-bisphosphate? Now this fructose 1,6-bisphosphate molecule is there. So here is one phosphate group attached. So this is a first carbon. This is a 6th carbon. Fructose 1,6-bisphosphate. If this substrate come in contact with this aldolase enzyme, what happens is that fructose 1,6-bisphosphate get converted into the glyceraldehyde 3-phosphate. So this is a glyceraldehyde 3-phosphate. So glyceraldehyde, again this is a aldehyde. You can see this is aldehyde group is there, C double bond O and H at the terminal position, and this is a glyceraldehyde 3 carbon atom, 1 2 3, okay, and 3 phosphate. At the third position there is a phosphate group attached. That is why we call it as a glyceraldehyde 3-phosphate. And another molecule is formed, we call it as a dihydroxyacetone phosphate. So this is a dihydroxyacetone. So this is the acetone group, C double bond O, and here phosphate is attached at the one position and there OH at the third carbon position. Okay, so this is a dihydroxyacetone phosphate. So now what is happening is that here with the help of the aldolase enzyme, so we have 6 carbon glucose molecule. This molecule got converted into the fructose 1,6-bisphosphate in the preparatory step. Now we have cleavage step. For the cleavage step, we want our 6 carbon glucose to get converted into the 2 compound. Which compound we want? We want 2 pyruvate molecule. And how this pyruvate molecule is forming? At the first step, the fructose 1,6-bisphosphate with the help of aldolase is getting converted into the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So basically 6 carbon molecules split into 2 3 carbon molecule, aldose and ketose. You can see glyceraldehyde 3-phosphate is aldose and dihydroxyacetone phosphate is a ketose, and this energy involved is a plus 5.73 kilocalorie per mole. Now we know that this glyceraldehyde 3-phosphate is involved in the downstream reactions of the glycolysis. Now what about this compound dihydroxyacetone phosphate? So there is one enzyme triose phosphate isomerase. So this one enzyme is there. What this enzyme does is that dihydroxyacetone phosphate get converted into the glyceraldehyde 3-phosphate. So this molecule can get converted into this glyceraldehyde 3-phosphate. So where we saw glyceraldehyde 3-phosphate, here it was glyceraldehyde 3-phosphate. So basically, with the triose phosphate isomerase, this molecule can get converted into this molecule or vice versa. So this dihydroxyacetone phosphate is getting converted into the glyceraldehyde 3-phosphate. Okay, let's see here. Glyceraldehyde 3-phosphate is forming. At the equilibrium, most mixture exists as a dihydroxyacetone phosphate. So at the equilibrium, most is in the dihydroxyacetone phosphate, and this reaction energy is a plus 1.83 kilocalorie per mole. So now we have from the glucose molecule how many glyceraldehyde 3-phosphate we got? We got 2 glyceraldehyde. So this one is a first glyceraldehyde 3-phosphate, and one dihydroxyacetone phosphate got converted into the glyceraldehyde 3-phosphate. It means that we are getting total 2 glyceraldehyde 3-phosphate. Okay. So these are some triose phosphate isomerase. So basically, when we say enzyme reaction is happening, means for example, this dihydroxyacetone phosphate is there. Enzyme is made up of the protein molecule. Protein molecule is made up of the amino acid. So for example, this one amino acid is there. Second, this is a glutamate is there. Histidine amino acid is there. Some reaction is happening. Some electron movement is happening. Enediol intermediate is happening, and then finally we are getting glyceraldehyde 3-phosphate. Okay, so whenever enzymatic reaction is there, when we say some enzymatic reaction is occurring, so basically what is happening in that enzymatic reaction? So enzyme is made up of protein molecules. So when protein molecule is there, like this molecule is there, so this molecule is there, this is a protein molecule. It will some amino acid will be present here, other amino acid will be present here, more amino acid can be present at position. But once our substrate is there, for example, this dihydroxyacetone phosphate is there, it is getting attached with this active site. So this is active site for this one, and all the reaction in which this glycerol dihydroxyacetone phosphate has to get converted into the glyceraldehyde 3-phosphate occurs at the active site. So that is the meaning of the enzyme. So that is what enzyme facility means. Enzyme is helping to convert this dihydroxyacetone phosphate to the glyceraldehyde 3-phosphate, and the name of the enzyme is a triose phosphate isomerase. Enzyme names, when we say enzyme name, it is also dependent on the which substrate is there. Triose means 3 carbon phosphate. 3 carbon is there, there is a phosphate is there. So 3 carbon containing compound which has a phosphate, it get isomerized, means it get converted, isomerization, ketone form is getting converted into the aldehyde form. That is isomerization reaction in this case. Okay, so this is a triose phosphate isomerase. Now glyceraldehyde. So again, talking with the glycolysis, we have first glucose. Glucose get converted into glucose 6-phosphate, glucose 6-phosphate to fructose 6-phosphate, fructose 6-phosphate to fructose 1,6-bis-phosphate, and fructose 1,6-bis-phosphate got converted into the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. DHAP again got converted into the glyceraldehyde 3-phosphate. So basically, we got 2 molecules of this. Here we are concerning the first single reaction. So glyceraldehyde 3-phosphate is there. Now this is a glyceraldehyde. So here aldehyde group is there, C double bond OH. At the 3rd carbon position, phosphate is group attached. Is attached. So that is why we call it as a glyceraldehyde 3-phosphate. And here we have nicotinamide adenine dinucleotide, NAD plus molecule, and the inorganic phosphate. So what is happening here? Enzyme involved is a glyceraldehyde 3-phosphate dehydrogenase. So whenever under biochemistry you are seeing some reaction, so you should pay attention to the substrate and the enzyme name. What is substrate name? Enzyme name? Glyceraldehyde 3-phosphate, because this is a substrate molecule, glyceraldehyde 3-phosphate, and dehydrogenation reaction. So dehydrogenation means there is a involvement of oxidation and reduction process. So what oxidation reduction is happening? So you can see here this glyceraldehyde is getting converted into the 1,3-phosphoglycerate. Okay, so this phosphate group, inorganic phosphate, is attaching here. Okay, so this is a 1,3-bis phosphoglycerate, and this NAD is getting converted into NADH. Okay, so this is a NADH. So this NADH can be a source of energy. We know that one NADH can give around 2.5 ATP molecule. Okay. So this is NADH molecule. It can be source of energy. If this NADH undergo the electron transport chain, under the electron transport chain, so this NADH can give 2.5 of the 3 ATP molecule. Okay. So this is a redox reaction. Redox means reduction oxidation reaction. Energy from the redox is used to form the acyl phosphate. Okay. From this glyceraldehyde 3-phosphate molecule, we have got 1,3-bis phosphoglycerate molecule, and the energy is involved is the plus 1.5 kilocalorie per mole. Okay, so this is basically consists of two coupled reaction. So what is happening is that under this reaction first, so basically this same reaction as a coupled reaction, glyceraldehyde 3-phosphate is shown here, and how ultimately first NADH is produced, and how this phosphate group is getting attached in the later position, and now we are getting this molecule 1,3-bis phosphoglycerate. Okay, so whenever you see this complex structure like this, so the structure looks complex, but if you pay close attention to the structure, basically this is all the meaning of the names will be in the structure, like 1 and 3 position is there. 1,3-bis phosphoglycerate means at the 2 positions phosphate group is there, so that is why we called it as a 1,3-bis phosphoglycerate molecule. Okay, so this is a 1,3-bis phosphoglycerate. Till now we have come till the 1,3-bis phosphoglycerate, and now we say purpose of glycolysis is energy. So you can see here energy is there. Okay, NADH. So this one is a source of energy here. So energy we have got here. If you allow it to undergo the electron transport chain as oxidative phosphorylation, it will give energy. Okay, 1,3-bis phosphoglycerate. Now what to do with 1,3-bis phosphoglycerate? So we have 1,3-bis phosphoglycerate molecule here. This is a 1-phosphate group at the first carbon position and third carbon position. That is why we are calling it as a 1,3-bis phosphoglycerate. Now we have ADP molecule. We see here ADP is there, adenosine diphosphate. When these two molecules come in contact in the presence of phosphoglycerate kinase, what is happening? One phosphate group is getting attached. This phosphate group is getting attached with this ADP molecule. Right now, adenosine diphosphate means adenosine containing 2 phosphate molecule. Okay, di means 2. One phosphate group more needed to make it ATP. So it is taking to the phosphate group from the 1,3-bis phosphoglycerate. Now it has taken this phosphate group, and now it has got converted into the ATP molecule. And what happens to this 1,3-bis phosphoglycerate? It gets converted into the 3-phosphoglycerate. Okay, this phosphate group, it has loosened to this ADP molecule, and ATP molecule we got. Now this is a 3-phosphoglycerate. So whenever 1,3-bis phosphoglycerate is there in presence of adenosine diphosphate, and in the presence of phosphoglycerate kinase molecule, this 1,3-bis phosphoglycerate get converted into the 3-phosphoglycerate. Okay, so this is a 3-phosphoglycerate, and ATP molecule is also formed in the same process. Okay, so now we have got energy here. So this is a purpose of our. So we have strict purpose. We want energy. Now we got energy. Okay, so formation of the ATP. So we call it as a substrate level phosphorylation. So if somebody asks what is the example of chemical reaction of the substrate level phosphorylation in the glycolysis, so you have to tell 1,3-bis phosphoglycerate get converted into the 3-phosphoglycerate in the presence of ADP and with the help of the phosphoglycerate kinase. So this phosphoglycerate kinase reaction is the substrate level phosphorylation reaction for the glycolysis. Okay, here we are getting substrate level phosphorylation. So this ATP molecule has formed directly in this process. Now we got 3-phosphoglycerate. Now what to do with this 3-phosphoglycerate molecule? we have 3-phosphoglycerate. Why we are calling it as a 3-phosphoglycerate? Because this phosphate group is attached at the third carbon position in this glycerate molecule, right? Now we don't want this phosphorus group at the third carbon position. For example, we want it at the second carbon position. So there is one enzyme, phosphoglycerate mutase. So what phosphoglycerate mutase does is that it takes this phosphate group and it say, okay, we will attach it here. So at the second carbon position, it got attached. First carbon, second carbon, third carbon. Now you can see it is at the second carbon. So this reaction of converting 3-phosphoglycerate to 2-phosphoglycerate happens with the help of the enzyme phosphoglycerate mutase. So this enzyme is involved, phosphoglycerate mutase, and it converts the 3-phosphoglycerate molecule to the 2-phosphoglycerate molecule. Now we got 2-phosphoglycerate molecule. Okay. Now what to do with this 2-phosphoglycerate molecule? There is a next enzyme involved, enolase. What enolase does is that it removes the carbon of this water molecule, H2O molecule, from this 2-phosphoglycerate molecule, and this get converted into the phosphoenol pyruvate. So you can see here this carbon is a single bond, here it is a double bond. So this is a enolic group. So 2-phosphoglycerate get converted into the phosphoenol pyruvate with the help of enolase, and during this reaction the water molecule gets out. Okay, till now we have reached to the point phosphoenol pyruvate. Now what to do with this phosphoenol pyruvate molecule? We have phosphoenol pyruvate molecule and we have enzyme phosphopyruvate kinase. With the help of pyruvate kinase, again there is involvement of ADP here, adenosine diphosphate, and now this ADP is getting converted into ATP. So this phosphate, we don't want this phosphate, you attach it with this ADP molecule, adenosine diphosphate molecule. Now this adenosine diphosphate get converted into the ATP, adenosine triphosphate molecule, and there is a conversion of ATP. Again, our aim is to get ATP. We got ATP here. What happens to this phosphoenol pyruvate? This phosphate group get removed and we got here pyruvate molecule. So phosphoenol pyruvate get converted into the pyruvate molecule with the help of the enzyme pyruvate kinase, and there is the involvement of adenosine diphosphate in which ATP is also formed in this reaction. Okay. So here the weakened ship, phosphoryl ship uses 2,3-bisphosphoglycerate. Then this energy is energy involved is a plus 1.1 kilocalorie per mole. If dehydration reaction, so this one is first was a phosphoryl shift. So phosphoryl shift means this phosphate shift from this third carbon position to second carbon position. That is a phosphoryl shift. It used this 2,3-bisphosphoglycerate molecule. Next, dehydration. The enolase reaction involved the dehydration, and during the dehydration reaction, there is a involvement of 0.44 kilocalorie per mole energy. Next is a phosphoryl group transfer. So phosphoryl group transfer. So this phosphoenol pyruvate molecule is there, phosphate is group there, ADP is there, and from two are combining and we are getting ATP molecule. That is what we call as a phosphoryl transfer. In the phosphoryl transfer, free energy involved is a minus 7.5 kilocalorie per mole. Okay, so here we are showing three reactions. Phosphoryl shift, first one, and then first one means 3-phosphoglycerate to 2-phosphoglycerate. Next, dehydration, 2-phosphoglycerate to phosphoenol pyruvate. Third reaction, phosphoryl transfer, phosphoenol pyruvate to pyruvate. So that is a phosphoryl transfer reaction. Okay, so these are the stages of glycolysis. So you can see here, first reaction, glucose 2. So all reaction we are seeing all together, right? Here on this page, glucose, we started with the glucose molecule. Glucose get converted into glucose 6-phosphate. Okay, here energy was involved. ATP, enzyme is a hexokinase, reaction was a glycolysis, and these are the different energy involved. We are already talking this one. This glucose 6-phosphate got converted. Glucose 6-phosphate enzyme was a phosphoglucose isomerase. This is isomerization reaction. Fructose 6-phosphate in the presence of ATP got converted into the fructose 1,6-bisphosphate, and ADP formed, and this reaction is catalyzed by phosphofructokinase reaction. Then fructose 1,6-bis phosphate got converted into DHAP, dihydroxyacetone phosphate, and glyceraldehyde 3-phosphate with the aldolase. That is the aldol cleavage reaction. Okay, and these three steps, they make the preparatory step of glycolysis. Okay, these are the post preparatory step. Okay, next DHAP get converted into the glyceraldehyde 3-phosphate with the help of the triose phosphate isomerase, that is isomerization reaction. Then glyceraldehyde 3-phosphate in the presence of inorganic phosphate and NAD plus get converted into the 1,3-bis phosphoglycerate plus NADH, that is a reaction catalyzed by the glyceraldehyde 3-phosphate, and this is a phosphoryl coupled oxidation. Next, 1,3-bis phosphoglycerate got converted into the 3-phosphoglycerate in the phosphoglycerate kinase. So you can see kinase, whenever kinase we are saying, it means that phosphoryl shift. You can see first kinase reaction, second kinase, it means phosphoryl shift there. Then phosphoglycerate kinase converted into the 2-phosphoglycerate, that is a mutase reaction, phosphoglycerate mutase. Then 2-phosphoglycerate got converted into the phosphoenol pyruvate with the help of enolase reaction. Then phosphoenol pyruvate got converted into the pyruvate with the kinase reaction. Again, kinase means phosphoryl shift is there. So these are the different steps. So you can count the number of ATP formed here. Okay, how many ATP were used? You can see here 1 ATP was used, 1 ATP was used. Total 2 ATP was used. How many direct ATP formed? You can see here 1 ATP formed, 1 ATP, 2. But if you notice here, DHAP, dihydroxyacetone phosphate, is getting converted into the glyceraldehyde 3-phosphate. Means the glyceraldehyde 3-phosphate can again undergo conversion into the pyruvate. So basically this is 2 ATP here, 2 ATP here. So directly how many ATP forms? 4 ATP form. And net, if we ask you net how many ATP are formed, net directly. So you can see these are two got expended. 2 रुपये होते आपल्याकडे, 2 rupees were there. First we invested 2 rupees. Now we got 4 rupees. 1, 2, 3, 4. So how many total rupees we have? 2 rupees. Okay, so that this is how you do calculation. So the total there are 2 ATPs. Now, for example, some other financial instrument you have, we call it as a NADH. So this energy molecule is there. NADH can get converted into the ATP, 3 ATP molecule. So how many ATP are there here? If you count this one, so again, if you consider this 3 ATP, this 3 plus 2, so there are the total 5 ATP. This 5 ATP can be there. Okay, so like that you have to calculate based on. So we have to write the reactions, and then you have to see one by one total expenditure, total ATP involved. Okay, so that was the fate of the glucose molecule. Glucose molecule was there. We have different enzyme, we have different substrate molecules involved, and there are some structural changes happening. So glucose molecule is getting attached with phosphate molecule, then it is breaking into the 2 carbon compound, and then 2 carbon compound is ultimately getting converted into this pyruvate molecule. Okay, now what can happen with this pyruvate molecule? So pyruvate molecule can undergo conversion into the acetaldehyde. If you are removing the carbon dioxide from the pyruvate molecule and acetaldehyde is forming in the first reaction, then again this acetaldehyde undergoes the conversion to the ethanol, and here NADH get converted into NAD plus. So this is a ethanol formation. This is how alcohol is produced, right? Ethanol molecule. It happens in the presence of the yeast. So yeast have this reaction to convert the pyruvate molecule to the ethanol. So under anaerobic condition, if this reaction is happening, pyruvate molecule is there. Glucose was there first. It got converted into pyruvate, and then pyruvate got converted into the acetaldehyde and the ethanol. Okay, finally we are getting ethanol. So this is one reaction. So next reaction is that this pyruvate gets converted into the acetyl coenzyme A. Acetyl coenzyme A can undergo further oxidation, and after further oxidation, it can give the more ATP molecules. So that is why we have more cycle, like TCA cycle, Krebs cycle. Okay, so in that one, the pyruvate is losing one carbon dioxide molecule and we are getting acetyl coenzyme A, or what is happening, sometimes muscle are exercising very fast, and in that case this pyruvate get converted into the lactate molecule. So this is a lactate molecule. Under this lactate molecule, what is happening is that NADH is getting converted into NAD plus, and we are getting lactate. That is why sometimes when people exercise very much, sometimes they get pain in their legs. So that is because of the accumulation of the lactate. Lactate is not getting time to get converted into energy, this oxidative process. So we are getting this lactate. So this is fate of the pyruvate molecule. What can happen to the pyruvate molecule? Pyruvate molecule can get converted into acetaldehyde and finally ethanol, or pyruvate can give the lactate, or pyruvate can give the acetyl coenzyme A. So these are the fates of the pyruvate. So when we say we were talking about alcohol production, ethanol. So what is happening? Which organism carry out this process? So yeast and some microorganism, they can carry out the formation of the ethanol. So we have the PDC require the thymine phosphate as a coenzyme. So pyruvate dehydrogenase enzyme is there. Decarboxylase reaction is there. Okay, PDC. So this is a first step. Now we have pyruvate molecule, and now we want to produce from this pyruvate ethanol. So how to produce it? So you have this pyruvate molecule here. Now first enzyme involved is the pyruvate decarboxylase. What is meaning of this enzyme pyruvate decarboxylase? Okay, substrate name is a pyruvate. That is why pyruvate decarboxylase. Decarboxylase means carbon dioxide is getting removed in the reaction. So you can see here CO2 is getting removed here. So we call it as a decarboxylation. D means it is getting removed. Carboxyl CO2 is getting removed. That is why we call it as a pyruvate decarboxylase. So this is a pyruvate decarboxylase reaction, and under this reaction, the pyruvate get converted into the acetaldehyde. Okay, this pyruvate is a 3-carbon compound. 1, 2, 3. Now this carbon dioxide is getting removed. Okay, so now this carbon dioxide got removed. How many carbon will remain? 1, 2 carbon, and this is a aldehyde group. So we call it as a acetaldehyde. Okay, so with the help of pyruvate decarboxylase, pyruvate got converted into the acetaldehyde. Carbon dioxide got removed. Now what to do with this acetaldehyde? Again, if acetaldehyde is there, we have one enzyme, alcohol dehydrogenase. What does this enzyme do? This alcohol dehydrogenase reaction to oxidation reduction reaction in our body. Oxidation reduction means some hydrogen is getting added at the particular position and oxygen is getting removed. Means hydrogen addition or removal, or oxygen addition and removal, that is the oxidation redox reaction, reduction oxidation. So acetaldehyde is there. Now NADH is there. So what is happening? This hydrogen group is getting attached here, and we are getting ethanol. So acetaldehyde get converted into the ethanol molecule. Now from acetaldehyde we got ethanol. What happened with this NADH? Once it loses the hydrogen atom, we got NAD plus molecule. So this is this molecule. So that is the alcohol fermentation. So the alcohol, what alcohol is producing, it is producing in the process like this process. Yeast molecule is there, glucose is there, glucose is undergoing the glycolysis. Okay, glycolysis happened. What happened with the glucose? Glucose get converted into the pyruvate molecule. What happened with the pyruvate? Again, yeast has an enzyme, pyruvate decarboxylase, which is removing the carbon dioxide. Carbon dioxide got removed. What happened to pyruvate? Acetaldehyde form. Okay, what happened to acetaldehyde? Again, yeast has an enzyme, alcohol dehydrogenase. In the presence of NADH, acetaldehyde is getting converted into the ethanol molecule. That is how we get this ethanol. Okay, so sometimes people drink this as a beverages like ethanol. So this is the production pathway: pyruvate to acetaldehyde, acetaldehyde to ethanol. Okay, so lactate fermentation. So lactic acid fermentation. So one phase we saw, pyruvate is getting converted into the ethanol molecule. Now second phase, how do lactate happen? In the previous step we saw pyruvate can get converted into lactate. So this occur in the muscle and microorganism, and this help in the regeneration of NADH. So we have lactate dehydrogenase enzyme present in us. So if pyruvate is there, the NADH in the presence of NADH, this pyruvate is getting converted into the lactate molecule. You can see here first carbon, second carbon, third carbon. So this is a pyruvate, 3-carbon pyruvate molecule. Okay. At the second carbon, OA double bond O is present, but here NADH carries hydrogen. So it says, okay, I will give hydrogen here. So here hydrogen it gives. You can see here. So it get converted into OH. So basically this pyruvate get converted into the lactate molecule. So this is how pyruvate get converted into lactate molecule with the help of the enzyme lactate dehydrogenase, and the molecule involved is the NADH, nicotinamide adenine dinucleotide. So NADH molecule. So NAD plus and dehydrogenases. Okay, he could have involved asतात here this are involved like in this reaction, first reaction. So this is the type of reaction, lactate dehydrogenase. So various dehydrogenases have the similar binding domain for the NAD plus, showing their common origin. So whenever this NADH nicotinamide binding domains are present, so dehydrogenase is, whenever dehydrogenases are there, they can have the similar binding domain for the NADH. So NADH, for binding with NADH, there is a Rossmann fold. So this Rossmann fold is present in the NADH where NADH bind. Okay, so this one is a NADH and this one is a nicotinamide binding half. Okay, with this here, this at this side they binds. Okay, so dehydrogenase is Rossmann fold. So now other molecules. So we saw glucose molecule can get converted into the pyruvate molecule. What about fructose molecule? So fructose molecule is there. With the help of fructokinase, fructose molecule get converted into the fructose 1-phosphate molecule, and the involvement of ATP is there. ATP get converted into ADP. Now fructose 1-phosphate molecule get converted into the glyceraldehyde with the help of fructose 1-phosphate aldolase. Fructose 1-phosphate aldolase nanta glyceraldehyde is forming and dihydroxyacetone phosphate is forming. Glyceraldehyde with the triose kinase get converted into the glyceraldehyde 3-phosphate. Okay, so that is how glyceraldehyde 3-phosphate is formed. And you all know that glyceraldehyde 3-phosphate in the second phase of the reaction after preparatory phase, it can give the ATP molecule, NADH molecule. So it is directly involved in the glycolysis. In that way, the fructose molecule can also a part of glycolysis, but two different enzymes are involved, fructokinase and the fructose 1-phosphate aldolase. Glycolysis. To use the galactose, it may get converted into the glucose 6-phosphate. Okay, now we have another molecule, 6-carbon galactose molecule. Okay, so this is a galactose molecule. We need a galactokinase. Kinase is involved means ATP phosphate molecule has to get incorporated into galactose. After galactokinase reaction, we get formation of the galactose 1-phosphate. Okay, so galactose is getting converted into galactose 1-phosphate molecule. Now what happens with the galactose 1-phosphate? We have UDP glucose and galactose 1-phosphate. Now galactose 1-phosphate uridyl transferase reaction happen where we get glucose 1-phosphate and the UDP galactose. Okay, UDP galactose for epimerase convert this UDP galactose to UDP glucose, and this can again help in the conversion. So that is here we are getting glucose 1-phosphate here. Okay, so basically glucose has to get converted into galactose. So some people have lactose intolerance. So what is lactose intolerance? So some people, they have deficiency of the lactase enzyme. So lactose is there. Lactose is a 2-carbon sugar. We call 2-carbon sugar here disaccharide. It is made up of the galactose and the glucose. So left is a galactose and right is a glucose. In the presence of hydrolysis reaction happen with the lactase, and they both get separated here. Galactose and the glucose can get separated. Okay, So what is galactosemia? It is inability to mobilize the galactose, and the missing the galactose 1-phosphate uridyl transferase. So liver disease, development of cataract and CNS malfunction can cause the galactosemia. Control of glycolysis. So at what step? So glycolysis is formation. Glycolysis leads to formation of ATP. But can we control it? So any biochemical reaction if we see, it needs check and balances. So control of what value glycolysis for the cell. So it provides the energy in form of ATP. So purpose of glycolysis is that it can provide the energy in form of ATP. So if you want to move your muscle, you will need energy in form of ATP. From where it comes from the glycolysis, for example. It provides the building blocks for the synthetic reaction. Okay, for some synthetic reaction like pyruvate is getting converted into finally acetyl coenzyme A. Acetyl coenzyme A help in formation of the some intermediates of the TCA cycle, and this TCA cycle intermediates can help in the formation of the some amino acid, and amino acid can help in the formation of the muscle. So it can help serve the purpose as a building block for the synthetic reaction. So what are the most common control point found? So enzymes that catalyze the irreversible reaction are the hexokinase, the first reaction named phosphofructokinase, and the pyruvate kinase. So if you see the reaction of glycolysis, sometimes arrow is pointing in the both direction, downward direction as well as the upward reaction. We call this reaction as a reversible reaction. But the certain reaction like first reaction of hexokinase or the phosphofructokinase or the pyruvate kinase, these are the irreversible reaction. Irreversible reaction means they don't reverse. If arrow is there, it is pointing upward to downward, not in the opposite direction. Okay, so these are the irreversible reaction. Hexokinase, phosphofructokinase, pyruvate kinase. Phosphofructokinase. Most important control point in the mammalian glycolytic pathway, the allosteric enzyme. This phosphofructokinase get activated with the help of AMP and the fructose 2,6 bisphosphate, and it get inhibited with the help of ATP, citrate, and the fatty acid. And this is phosphofructokinase. So relative velocity we can calculate how much concentration fructose 6 phosphate concentration, and relative comparison we can do with the help of low ATP. Reaction will go like this. If high ATP is there, reaction will be slow. That is why we call it as a inhibitor by the ATP molecule. You can see here, low ATP is there, reaction is faster. Okay, reaction velocity is faster, and if this ATP is high, that reaction is comparatively slower. Okay, so phosphofructokinase, hexokinase. Hexokinase is inhibited by its product glucose 6-phosphate. So sometimes there is a negative feedback inhibition, means glucose is getting converted into the glucose 6-phosphate with the hexokinase, but glucose 6-phosphate, which is formed in the hexokinase reaction itself, it may inhibit this hexokinase reaction. Okay, so this glucose remains in the blood. Glucokinase is the isoenzyme of the hexokinase, and it is not inhibited by the glucose 6-phosphate. So there is another enzyme, liver. Liver continuously has convert, for example, glucose to glucose 6-phosphate, and now glucose 6-phosphate is accumulated, and we are saying hexokinase is getting inhibited. So liver has devised another isoenzyme, glucokinase, which serves a similar function to convert glucose to glucose 6-phosphate, but it is not getting inhibited by this end product, glucose 6-phosphate. It has a lower affinity for the glucose. Okay, so there are isoenzymes are there. Isoenzymes means basically this enzyme catalyze the similar reaction, but they have some differences, structural differences in there. Okay, hexokinase. So pyruvate kinase is there. So pyruvate kinase exists as an isoenzyme. EL form is there. It is predominant in the liver. EM form of pyruvate kinase is mostly is in the present in the muscle and the brain. Pyruvate kinase is an allosteric enzyme. It is activated by the fructose 1,6 bisphosphate. So pyruvate kinase is there. If you want to activate, you can this molecule, if it is there, fructose 1,6 bisphosphate, it get activated. If ATP or alanine is there, then this pyruvate kinase can get inhibited. Okay, so EL form, EL for liver, EM form, EM for muscle, and EM form is also present in the brain also. EL form is influenced by covalent modification, and this EL form, which is present in the liver, is inhibited by the phosphorylation. Okay, so what happens is that pyruvate kinase is there. If you dephosphorylate, means if you remove the phosphate from this enzyme, pyruvate kinase, then it is in the most active form. Then this phosphoenol pyruvate can get converted into the pyruvate and the ATP molecule. But in the presence of ATP, what is happening? Okay, this pyruvate kinase is getting attached with this phosphate group. Now this is a phosphorylated pyruvate kinase, and now this one is a less active. It has a less activity. So most active form is a dephosphorylated form. So the condition in which the ATP is less, in that case, this enzyme is active. If ATP is more in the body, then this enzyme is inactive. So already ATP in the form of energy is there. We don't want to produce more energy. So what ATP is doing? It is phosphorylating this pyruvate kinase. If fructose 1,6-bisphosphate is there, it is positively influencing this enzyme and it is producing more pyruvate. But ATP, alanine, if it is there, it is inhibiting this pyruvate kinase enzyme. So glucose transport. So when we say glucose is there in the blood. Now it has to cross the cell membrane and it has to get inside the cell. That is called as the movement of the glucose from one transport. We say transport. Blood, inside the blood, glucose is there. This glucose has to get transported inside the cell. That is a transport. Like we are going from Parvani to Nanded. So that is a transport from one location to another location. So similar way, glucose is there. Glucose has to go from the blood location into the inside the cell location. For that, glucose transporters are needed. Okay, so what this transporter does is that they facilitate the movement of the glucose across a membrane. And what kind of the molecule is the transporter? So when we say the transporter molecule is a small protein embedded in the plasma membrane. So basically they are made up of the protein molecules, so embedded in the plasma membrane. For next slide, for example, these are the mammalian glucose transporters. So this one is a cell membrane, and you can see here protein. So it is a intramembrane protein, and this is a membrane spanning. So this protein is spanning like this. So basically these are the glucose transporter. Okay, you can see here amino group is there, carboxyl group is there. So glucose transporter, they can be different types. Okay, so GLUT1 we said as a glucose transporter. Now we want to give it name. So what name do we give? We give for glucose GLUT, GLU for glucose, and T for transporter. So that is why we gave name GLUT1. If you see all mammalian tissue, this GLUT1 is present, and Km for a Km affinity constant is a 1 millimolar, and its function is the basic glucose uptake. GLUT2 receptor present in the liver and pancreatic beta cell. Km is a 15 to 20 millimolar. In the pancreas, it plays a role in the regulation of insulin. In liver, it removes the excess glucose from the blood. Now there is another transporter, GLUT3. It is present in all mammalian tissue. Its Km is a 1 millimolar and function is the basal glucose uptake. GLUT4, it is present in the muscle and fat tissue. Km is a 5 millimolar and in the muscle plasma memory increase with the endurance training. So if you are doing more exercise, then this GLUT4 receptor increases. GLUT5 receptor is present in the intestine. Or in intestine also we want glucose to get into the intestinal cell, and its primary function is the fructose transport. Okay. Glycolysis and the cancer. So there is some tissue which are growing independently without any control. We call them as a cancerous tissue. Okay, if somebody is getting tumor on the hand, then muscle mass is growing at very faster rate. We call it as a tumor. So why are rapidly growing tumor cells depending on glycolysis? So this tumor cell needs for energy they depends on the glycolysis. So what is happening is that tissue is growing so faster at the particular location that there is an insufficient oxygen supply. So body who supply the oxygen to the different location in the body, hemoglobin supply the oxygen, hemoglobin combining with the oxygen molecule. But as the tumor cell is growing so faster and faster, this tumor cell cannot meet the need of oxygen. So that is why for energy supply, they depends on the anaerobic glycolysis. So HIP is there, hypoxia-inducing transcription factor that stimulate the Mary glycolytic enzyme and the GLUT1 and 3. It also stimulate the vesicular endothelial growth factor VEGF. Okay, vesicular endothelial growth factor is also stimulated. Okay. So that is about the glycolysis. So this is how glycolysis reactions have happened. Okay, so today we will only learn till glycolysis. Okay, gluconeogenesis we will learn in the next lecture. Okay.